Okay, thank you very much for inviting me to talk to the British Humanist Association, especially on this day. It is a great privilege. It is also a great privilege to be introduced by my old tutor, as he has just so generously described. He's kind of a tough act to follow. Steve was, in fact, one of the reasons why I chose genetics as a subject, and was also one of the reasons why I chose to become a writer about genetics and evolution. Unfortunately, he has done most of the jokes about genetics and evolution. So in this talk, I decided to find a subject that he hasn't written about, and I just mentioned that to him in the green room beforehand, and he said that he had. So, as he says, all of the jokes that I'm about to do have probably come from him. But before I get too fawning about my old tutor, I do need to remind him that he left me, abandoned me, and 7 other students at Malaga Airport in 1994, after a field trip, having accidentally failed to tell us that the flight was an hour earlier than the one that he got on. Now, the two things that I'm going to talk about tonight are synthetic biology and the origin of life. Now, these are immensely complex subjects, and they're very unsettled. This is very current science. Now, over the years, I have witnessed many fairly furious debates between scientists. They are a quarrelsome bunch, but I've never come across more furious, more personal, and more bad-tempered debates than the fields of origin of life studies and synthetic biology. And I think that is because it is so unsettled. It also makes it very exciting. But it does mean that I can't necessarily offer you any answers tonight. But I will endeavour to give you a most serene and gentle overview. I think there are questions afterwards, and you may get furious with me at that point. Now, these are extraordinary times for biology. It's now 153 years since the publication of The Origin of Species by that man over there, although I think in real life he was about 3 inches taller than that. In that, as you all well know, he outlines the theory of evolution by natural selection, and that is a theory in the scientific sense of the word, meaning that it is an agglomeration of countless observations and experiments and pieces of data that come together to describe something which is effectively indistinguishable from fact, and whose wholesale replacement is now extremely unlikely. There is an overwhelming body of work which demonstrates the idea that via the mechanism of genetic mutation plus generational time is the reason why we have the great diversity of species that have existed on Earth. In terms of studying biology, this is an era where we're lucky enough to be filling in the gaps, to be filling in the details and testing this theory to destruction. Now, as a result of that, all of the best books about evolution have already been written, some of them by Steve and other people such as the absent Richard Dawkins. What I'm concerned about here, if I can employ sort of Hollywood terminology, is the prequel and the sequel to evolution. In physics, asking the question what happened before the Big Bang is effectively meaningless. But in biology, that is not the case. What happened before the first life form is a very interesting and scientific question. And now that we have a great understanding of how genetics and how life forms actually work, what we do with that information is equally important. But in order to get to that prequel and get to that sequel, we first have to look at what happened in the middle. So this is a picture that you'll all recognize. This is the first tree of life that Darwin drew. I almost said Dawkins there. That Darwin drew in his notebook in 1837. And while it's quite simple, it shows that species are the divergence from a common ancestor. This is what the picture looks like now, or in 2003. We estimate that there are about between 1.8 and 2 million species currently alive on Earth. But we also know that of all the species that have ever existed, more than 9 out of 10 of them are already extinct. So this is just a tiny sample to show that tree in its most recent form. And if you're interested about where we are in that, if you just look to the very top left, you are here. Except biology is something that is a subject that I over the last 17 years of studying it, have come to the conclusion gets more complex the more you look at it. The root of the tree of life is occupied by two domains of life, archaea and bacteria, both single-celled organisms, simple single-celled organisms. And they do something which the other domains of life don't do, and that is that they're capable of swapping genes between siblings, rather than, as we do, from parent to offspring. And what that means is the root of the tree of life is a little bit more like a thicket or bushy, rather than a tree. This is a model to show that tree. Now that is the complex interaction between bacteria and archaea at the beginning of life on Earth. That doesn't mean that Darwin was wrong, as the New Scientist rather naughtily put on their front cover on this day in 2009 on his 200th birthday. It meant that what it means is that Darwin was not wrong. The tree is still a very useful way of describing descent. Darwin had no knowledge of archaea and little knowledge of bacteria. He had no knowledge of genes or lateral gene transfer, which is the thing that we're talking about. So as a public service, there you go, I've fixed that for you. The truth of this picture is the culmination of three great landmarks in science. The three grand unifying theories of biology. One of them is, so by the end of the 19th century, two of those were in place. One of them is the theory of evolution, which I don't need to go into further, but can be summarized like this: descent with modification. But the first one came several years before that, and that is cell theory. Now, many European scientists were credited with coming up with cell theory. Mostly the credit goes to these two chaps on the left, Schwann and Schleiden. But equally important, though written out of history, was Robert Remak. Now, cell theory can be summarized in these two statements. All life is made of cells. We know of no life forms that are not cellular in their basis. And the second thing is that cells can only come from pre-existing cells. They are only born by the division of existing cells. Now, what that means is if you combine evolutionary theory with cell theory, two robust theories in the purely scientific sense, just like gravity, then you're unavoidably drawn to a point where the origin of life has to have come about from a single cell. And this introduces a concept that existed for a long time in culture, which I'm going to talk about for a bit. The most important thing for the purposes of our talk are that cell theory was the death knell for a concept called spontaneous generation, which was for more than 2,000 years the main way we thought the genesis of life occurred. Now, all cultures have creation myths. It is a really fundamental aspect of humanity to try and work out where we came from. Here are some. This one is one of my favorites. This is the Egyptian creator god Atum, who created himself, which is a sort of conundrum you can't really get round. But in order to create humanity, he indulged in the rare talent of autofellatio, and then spat out the first man and the first woman. Now, I know it's very childish to point out something so scatological, but it does make me kind of laugh. And it is a nice comparison with the relatively conservative story of Genesis, the creation story as told in the Bible, a very conservative Judeo-Christian story. Note the caption for later on, Adam left. That will become apparent why I've pointed that out in a bit. Now, this is an example of creation ex nihilo, out of nothing. And that is what spontaneous generation as a scientific term is. It is the creation of living matter out of inanimate matter. Aristotle was the first person to really describe what he believed was spontaneous generation after he claimed to have observed mollusks and certain types of fish undergoing spontaneous generation. And this is an idea that persisted in many different forms over many centuries. Here are some examples. I kind of like this slide because it's sort of like introducing two people with parallel names. You sort of go, goose, barnacle, barnacle, goose. The goose barnacle is a type of mollusk, and the barnacle goose is a type of goose. But in 11th century Christians asserted that the barnacle goose was spontaneously generated from the goose barnacle. It actually turned out that the reason they were doing this was not for any scientific purpose to describe the origin of life. It was so that they could eat geese during Lent, on the grounds that if the goose was spontaneously generated, then it was unnatural and immaculate, and therefore they could eat it. Pius III got hold of this piece of information and rapidly banned it. So over the course of the centuries, there are many other examples of spontaneous generation described in semi-serious pseudoscientific literature, such as rotting logs in lakes could mutate into crocodiles. There was an experiment by the father of chemistry where he sealed a sweaty shirt of his with some ears of corn and left them for 21 days, and then described that mice spontaneously emerged from that. And this idea even continues in our culture to this day. One of the descriptions of spontaneous generation was the emergence of bees from the carcasses of rotting lions, which is described in the Bible and still is on the label of Lyle's golden syrup. Now, spontaneous generation was finally killed in the middle of the 19th century with some good experimentation, some good science, finally during the process of the development of cell theory, but significantly with this very simple piece of equipment, the swan-necked flask by Louis Pasteur. He took one flask with some rich broth in it and left it open, and it became contaminated with bacteria and mold. And he took another flask and put this swan neck on it, still open to the air, but it didn't ever become contaminated because the bacteria or spores could not get through the swan neck. Now, moving on from spontaneous generation, it is Darwin Day, and so I think as it should be the tradition, we should all bow before what I believe are some of the greatest words written in English prose. The final paragraph of the final chapter of The Origin of Species. There is grandeur in this view of life with its several powers, having been originally breathed into a few forms or into one, and that, whilst this planet has gone on cycling according to the fixed law of gravity from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. The combination of cell theory and evolutionary theory leads us logically to the idea that there is just into one. There was a singular first form, and that this phrase is rendered even more beautiful, even though The Origin of Species does not discuss the origin of life itself. Now, we have a name for that entity, that original cell, and it is LUCA, the last universal common ancestor. And the study of origin of life biology is to determine what LUCA was and how it came to be. There are competing theories about this, and not all of them are equal. And I'm going to talk about three of them. The first is the one that we have to get out of the way quite quickly, and that is panspermia. This is the idea that has existed for a couple of centuries that life abounds in the rest of the universe and was delivered to Earth in meteorites. There is a sort of interesting backstory to this, which is that we believe that LUCA existed 3.9 billion years ago. And that coincides with the end of the most destructive period in the Earth's history, which is called the Late Heavy Bombardment, a period where meteorites rain down from the skies for several 100 million years with enough destructive power to vaporize the land and the sea on a monthly basis. Now, as soon as that ended, life began. Panspermia was taken seriously for a number of years by serious scientists, including people like Francis Crick and Fred Hoyle. But we can disregard it very easily and very quickly, and you all should, because there simply is no evidence for it whatsoever. We know of no other life forms other than what exists on Earth, and that is enough to rule currently panspermia to be in the realms of science fiction. I find it interesting that we get so excited about discoveries that determine that that things were delivered, that matter was delivered to Earth from space, because it sort of forgets the fact that we are an entity in space and that we were formed in space. This is an artist's depiction of the protoplanetary disk, the detritus after the infant sun was born. All of this matter is what the planets of the solar system are made of, so perhaps it's not that surprising that stuff from space is on Earth. That's not to say that there aren't very interesting, extremely thrilling things about the delivery of matter and potentially biological matter to Earth from space. This is a cross-section through a meteorite that landed in the Australian town of Murchison in 1969, just after the first moon landings. And a team at Imperial College a couple of years ago determined that this carbon-rich meteorite contained one of the letters of genetic code, uracil. But uracil, one letter of genetic code, that is not life. Now, incidentally, I've mentioned that the origin of species doesn't talk about the origin of life itself, and this is one of the arguments. I say arguments, foolish statements that creationists sometimes use in their arsenal, which sort of goes like this: evolution and the origin of life don't account for, evolution and the origin of species don't account for the origin of life, therefore Darwin was wrong. Now, this is an argument technique called whataboutery. You might have come across it. It's the introduction of a random and slightly irrelevant fact to destroy the first. But Darwin, being that clever chap that he was, he did actually kickstart the science of the origin of life in a letter that he wrote to his friend Joseph Hooker in 1871, in which he says, "But if," and oh, what a big if, absolutely dreadful handwriting that he had. I've held this letter in my hand, and I had to read it out for a TV program that I was making. And after 8 takes, because I couldn't read it, they decided to actually print it out for me. But if, and oh, what a big if, we could conceive of some warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity, etc. present, that a protein compound was chemically formed, ready to undergo still more complex changes. Now, Darwin was prefiguring what would become the most prevalent and dominant theory about the origin of life. 50 years later, independently, these two men, Alexander Oparin and JBS Haldane, how so? Both communists, strangely, although that is a random observation. Are they the wrong way around? Oh, see, this is what happens when you're introduced by a bloody tutor. I have never corrected you in a lecture once. Alexander Oparin and JBS Haldane applied a little bit more scientific rigor to this warm little pond idea. And they both speculated that if you've got the ingredients right, and if the atmosphere of the early Earth was right, then you would eventually see the spontaneous emergence of life forms. Haldane, that's him on the left, I'm told, he coined a phrase that has become the sort of meme, the successful way of describing this theory, and that is primordial soup, something which is ingrained in our culture now. If you get the ingredients right, you simmer for a geological time period, then life will emerge. The next great landmark in this primordial soup study was done by this man, Stanley Miller. The experiment was actually done when he was 22 years old as a graduate student. And what he did was to construct a set of glass apparatus, sealed glass apparatus, in which he puts the ingredients of what they thought at the time might have been present on the early Earth, which is ammonia, carbon dioxide, water, hydrogen, and methane, and heated it up and put an electrical spark across it and ran this for a couple of weeks. And within a few days, the liquid, the water in it, turned brown. And he analysed that using the techniques available in 1953 and discovered that he had produced this mixture had produced 5 amino acids being essential for all living things. There's a nice coda to this story, which is that those samples were rediscovered in this century, and in 2010, a modern analysis was done, and in fact they found 25 amino acids, which is more than the number of amino acids that have made up all of the proteins that have ever existed in living things for the last 4 billion years. But amino acids are not life. Now, this primordial soup idea mutated and matured over the next few years, and with the discovery of a type of molecule. Let me rephrase that and go straight onto this card. It matured and mutated into a theory which is now known as the RNA world hypothesis. And the reason for this is because RNA, which is DNA's cousin, has 2 roles. One is that it is informational. It contains a code just like DNA does, but also that it has some basic enzymatic functions. It can act as a catalyst. So the suggestion was that the first life forms were running off information derived from RNA, that it could actually process basic metabolism and contain information. Now, this field was given a great fillip in the last few years with the advent of these type of molecules, which are called ribozymes. Now, these are RNA enzymes that can self-catalyze their own replication. This work done a couple of years ago by Jerry Joyce and the University of California in San Diego put together a semi-random series of very short ribosomes in the right broth and fed them. And what happened was the ones that were most successful at replicating became the dominant species. He then introduced slight random variation within that dominant species and repeated the same experiment. And again, the ones that were most efficient were the ones that came to dominate that mix. Now, to any evolutionary biologist, that sounds a lot like natural selection, but on a very simple molecule. But is that life? Well, it's Darwinian, but I don't think that that is a life form. Here's another example. Cell membranes are absolutely inherent to cells. They have to be self-contained entities. And the formation of the cell membrane, which is a complex structure, is something that is still a bit of a mystery. Jack Szostak, who won a Nobel Prize in 2009 for some totally unrelated work, has put together a system using some very basic fatty molecules where spontaneously you get the formation of cellular-like structures with a membrane which is a bit like the types of membranes that we see in many life forms now. It also incorporates RNA into it, preferentially over other chemicals. Is it life? Well, I don't think it is. And so all of these ideas, which I think individually are fantastic pieces of research and outstanding pieces of science, all in this framework of origin of life under the terms of primordial soup or RNA world hypothesis, I think they don't really go far enough in addressing the question of what life is. So, well, what is life? And the answer is we don't have a definition of what life is. We're very good at recognizing it when we see it. This is what I find is most useful in describing what life is. Life is the opposite of decay. Everything in the universe is naturally predisposed to decay, but life doesn't do that. Life is an ordered state, and it's perpetually ordered through reproduction over 4 billion years, and always has been. We have to turn slightly to physics here, which is always a little bit galling, but basically what we're describing is the times arrow through the second law of thermodynamics. The universe is naturally predisposed to decaying, so eventually everything will be much more chaotic. But life manages to battle against that chaos and have self-contained order, and has maintained that order over the process of evolution for almost 4 billion years. Now, I find that quite an uplifting thought, that no matter how chaotic you think your life is, in scientific terms, you are a tiny, tiny pocket of order as the universe irreversibly descends into chaos. Now, that's some sort of grand-sounding rhetoric, which is actually really guff. But what does that mean experimentally? Well, it means experimentally that life is a process of energy capture. It is a process which essentially requires the ability to extract energy from the environment and to hang on to it long enough that it can perform its primary function, which is to reproduce. Where would such a situation occur? You cannot have genetics without energy input. You cannot have reproduction without energetic input. And so we're led, I think we are led irretrievably to think about life as a metabolic process fundamentally, and that reproduction and information exchange is subsequent. So all of those experiments which are about the transfer of information over generational time are actually subsequent to the capture of energy. Now, in the 1980s, a location on Earth was discovered where it has a lot of characteristics which fulfill this as a potential spot for the origin of life on Earth. This is a picture of what's known as a white smoker, an alkali hydrothermal submarine vent. It's in a place called the Lost City, which is somewhere in the mid-Atlantic. It wouldn't be lost if I knew exactly where it was. It's on the Atlantic Massif. And what these are these, as hydrogen and methane bubble up from the active Earth, they create a rock, and they bubble through rocks which become porous and create little pockets. Now those pockets sometimes they have metals on the inside walls of them, and those walls are predisposed to forming natural membranes. And so with hydrogen atoms bubbling up and potential membranes, you potentially have a system where you have a maintained energetic disequilibrium between the outside and the inside, and that is a fundamental constituent of metabolic life. Now, these places are very inaccessible and very difficult to get to, so it remains hypothetical until you do what scientists do best, which is to experimentally test it. At this point, we come to the very cutting edge of the research that is being done, to the extent that the experiment hasn't actually started. Now, this humble piece of equipment, rather Heath Robinson piece of equipment, is on a bench on Gower Street in the aptly named Darwin building at UCL, and has been built by Nick Lane, whose books you might have read. And what this is it is a bioreactor, which is a rather grand title for a pot with some bubbling liquids coming through it. Inside, I spoke to Nick yesterday, and they've finished the ceramic that is going to go in it, and they're happy with it. A porous ceramic material that will sit inside there as they bubble up different gases through different liquids and attempt through an outflow pipe at the top to analyze what sort of products are being made as those gases circulate around this porous material. As I say, he hasn't started yet. We await the results of this experiment imminently. But it strikes me that this is, appropriately enough, in the Darwin building on Gower Street, at the moment, experimentally, the most likely place that we will see, as near as we can, what could be described as the origin of life. We cannot repeat what happened 4 billion years ago because we don't know. But what he will be looking for in that experiment is basic metabolites, basic evidence of biochemistry that is emerging from this bubbling energetic situation. Now that's the end of part one. Part two is evolution, the sequel. So in order to get to this point, in order to talk about the origin of life, we've discussed the two of the cornerstones of biology, two of the grand unifying theories of biology. In order to get to the sequel, what happens next, we need to talk about the third. Now, this is a very well-told story. In 1953, Francis Crick on the right, James Watson on the left, used the data of Rosalind Franklin to determine that the structure of DNA was the iconic double helix, which you all know about. The double helix is a very clever molecule because it does two things. The internal rungs of the double helix are made up by 4 chemical letters, A, T, C, and G, and they pair up in a very specific way. The order of these letters is the code. This is the code that encodes proteins, that makes all of the things that living things rely on. But the way that they pair is very specific. A only pairs with T, and C only pairs with G. And that is what enables DNA's second clever function, which is that it is a perfect replicating system. If you split the 2 strands of the double helix apart, you have all the information on both strands to replace the missing strand. Every time you find an A, you put a T. Every time you find a C, you put a G, and so on. And this is the system of reproduction that all life has always relied on. We know of no organisms that don't use this very simple, basic, conservative system of 4 letters of DNA code that encode amino acids, of which life uses 20 to make up all the proteins that have ever existed in every life form as far as we know. Proteins, all life forms are made of or by proteins. And so this has become known as the central dogma. Now, dogma is not necessarily a useful word in science. It was Francis Crick who called it the central dogma, and he regretted that and was interviewed several times after he called it the central dogma in the 1970s. When asked, why did you call it dogma, he said, I didn't really know what dogma meant. Now, as a result of this universality of the genetic code, we have our understanding over the last 20 or 30 years of how genes, how DNA works, has led us to the point where we effectively can take DNA, take genes from any organism and put them into any other organism, because it is a universal language across all life forms. And there are some striking examples of this, which let me just give you a couple of examples. This is a goat that I met on a recent travel to a farm in Utah. It's Freckles, her name is. Now, Freckles has been genetically modified so that she produces spider silk in her milk. So when I met Freckles, I milked her, and we took the milk back to the lab, and we processed it to remove the fats, and then put a little bit of pressure on it in a special machine, and then I took a glass rod, and out of that liquid pulled a single thread of spider silk, which I then spooled 7 meters worth onto a spool. Spider silk has amazing physical properties. It is akin to steel, but is much more elastic. But it's much better than anything that we can manufacture. And you can't farm spiders because they're cannibalistic. You put 2 together, one will eat the other one. So this is a very desirable product for us to have. And this effectively is a form of extreme farming. We can produce something, we can breed an animal so it produces something that we want. The real difference between Freckles and traditional farming is that the species boundary has been crossed. We no longer are bound by crossing 2 animals that are very closely related to each other. Because of our ability to manipulate the universal genetic code, we can take spider genes and put them in a goat. Now, it's a little bit, it might seem a little bit vain to put a picture of yourself up in your own talk, but there is a specific reason for this. I wrote an article about Freckles and the spider goats for The Observer a couple of weeks ago, and they used this picture, and they felt the need to caption it with yeah. Just in case there was any doubt about that. Another very striking, visible example of our ability to genetically engineer, to genetically change organisms, comes in the form of the fluorescent animals that you might have seen on the news. This is not an engineered animal. This is a jellyfish that glows green in the dark ocean, and it does that because it has a gene which produces a protein that is fluorescent, and that protein is called green fluorescent protein, which is not the most imaginative title for a protein that fluoresces green. These mice have that gene being expressed in every single one of their cells because it's a very useful way of tagging genetic manipulation that we might have done experimentally that otherwise would have been cryptic or difficult to trace. And it's not just for fun. This is this fluorescent cat is from a paper last year that was created by AIDS researchers so that they could trace a particular protein that attacked the HIV virus. So not just for fun, incredibly useful. Now, you can't really talk about genetic engineering without eventually coming to this fellow. This is Craig Venter. Now, Craig Venter was a big cheese in the 2001 publications, plural, of the human genome sequence. His was a private venture. Last year, again, he made headlines for what he described as the creation of the first synthetic cell, which is Mycoplasma mycoides JCVI-SYN 1.0, which became known as Cynthia, which incidentally is something that you shouldn't Google at work. Now, what was Cynthia? Well, I don't think that she was synthetic. She? Look at all that anthropomorphizing. I don't think it was a synthetic cell. This is what he did. He took the genome from a bacteria with the smallest known genome, and input it into a computer. And then he had the computer synthesize that DNA in full with certain modifications. And they took that computer-synthesized DNA and put it into the eviscerated chassis of another closely related cell. So is it true that that was alive? Yes, I think that it is. Is it true that it didn't exist beforehand? Yes. It was not descended from an existing bacteria, but was it a synthetic life form? I don't think that it was. I think that what Craig Venter did was to copy and reboot life. Now, incidentally, in order to demonstrate that he had, this was a new creation made by his hand rather than a naturally occurring thing, Venter put in several coded messages into the genome of Cynthia, and some of them were a web address, and some were the names of the 42 scientists who were involved in this project. And also he put 3 quotations, apposite or pretentious quotations, depending on your point of view. If you like puncturing hubris, how about this? The first two. The first was from James Joyce: to live, to err, to fall, to triumph, to recreate life out of life, which seems extremely appropriate. The James Joyce estate are famously protective of their property, and within a week they issued a cease and desist writ for breach of copyright. The second quote: see things not as they are, but as they might be. And the third was this, by Richard Feynman: what I cannot build, I cannot understand. Now, any geneticist will tell you that accuracy in sequence is absolutely paramount. Accuracy in sequence is the difference between having a functional CFTR gene and having cystic fibrosis. The actual quote that was written on Richard Feynman's blackboard at his death, supposedly, was this: what I cannot create, I do not understand. I phoned up the Venter Institute and said, how did that happen? And the answer was, someone looked it up on the internet. Anyway, now there are some potentially contentious issues which may come up in the discussion. I'm not going to get into them here, but it's interesting to see how the press covered them. Now, I realise the Daily Mail is probably not the first place you'd go for sensible scientific discourse, but I think in this case they really outshone themselves. The headline was: scientist accused of playing God after creating artificial life by making designer microbe from scratch. But could it wipe out humanity? Now, apart from winning the award for the longest headline in history, I really think they've outdone themselves here. So I've taken the liberty of annotating this headline. Scientist accused by the Daily Mail of playing God, don't really know what that means, after creating artificial life, well, not really, by making designer microbe from scratch. Well, they definitely didn't do that. Could it wipe out humanity? No. As you were, you clever people. No, why no? Well, Mycoplasma mycoides is a goat pathogen. It's a bacteria that causes a minor form of mastitis in goat udders. There's a sort of goat theme going on here. It's not fatal to goats. Venter also took out the pathogenic bits and neutralized the pathogenic bits of the DNA in this goat pathogen, so that it couldn't even cause mastitis in goats. So really, no. Now, this is all in the realms of extreme genetic engineering. It shows our immense ability to manipulate the fundamental language of life. In the last few years, it's mutated and grown up, matured into a new science, which has become known as synthetic biology. Now, this is one description. It's a sort of loose, synthetic biology is a broad term, and this is one description of it. The application of engineering principles in the design and redesign of natural biological systems for useful purposes. For natural biological systems, we're talking about DNA. For useful purposes, well, that really is the engineering ethos, which has come before the much more experimental and scientific principles that have been determined by genetic engineering. Here are some examples. This is a type of cress that, with synthetic biological circuits introduced into it, has been designed such that when it grows in the presence of landmines, it goes purple. So in fields that have many landmines in them, you can scatter these synthetic biological plants, and where they go purple, you see a landmine. It makes them very easy to determine. I think one of the most significant advances in the scientific literature in this field came just last year, in September 2011, where a team from MIT designed a biological circuit, a team of computer scientists fused with molecular biologists, designed a circuit that would identify a specific type of cancer cell, HeLa cells, using a 5-point criteria, a logic flowchart. And if the recipient cell satisfied those 5 criteria, it would release a toxin which would destroy it. Now, this is the absolute obverse of the current cancer therapies that we have. This is an assassin cell that, unlike chemotherapy, which attacks many things indiscriminately, or radiotherapy, which does much the same, this is a cellular form of cancer therapy which attacks only cancerous cells. This works in vitro. It is about to be tested in animals. This is a figure from that paper, which I show not because I have any understanding of what's going on in it, but just to show quite how much biology has become like electrical circuitry. Now, all of the component parts in that experiment were sourced from a foundation called BioBricks. BioBricks run an online repository of currently 7,000 or 8,000 biological components, as they call them, or bits of DNA, as they were traditionally known. And they're freely available, which is part of this sort of democratic ethos that has emerged within this new field of synthetic biology. This is also demonstrated by the fact that every year they run a competition for the last 5 years, which is called iGEM, which stands for the International Genetically Engineered Machine Contest. And it is for undergraduates, and there's a separate competition for school children to use any of the available parts in the registry, in the standard parts registry, to design a problem, design a solution, and build it. And to exemplify the sort of engineering ethos behind this, this is the prize that they take home. It's called the golden brick, but it's actually silver. And the team that won it, 2011, last year, from Washington, they had designed a microbe that would produce a form of biodiesel which is as efficient as diesel. Now, I'm going to wrap up now so we can have some questions. There are, I'm sure, many questions. I have a pro-science agenda, and there are issues, some more genuine than others, and I'm sure that they will emerge in the discussion. But it's my contention that we are on the cusp of an industrial revolution. This is technology which has the potential, and already is, changing every aspect of human existence, from fuel production to drug production to energy consumption to environmental cleanup. And there are certainly issues within here. I believe that we, as a public, it is our responsibility to be informed about these new technologies, and rather than be fearful of them, use them in the best possible, most informed way for the betterment of humanity. And that's where I'll end. Thank you very much.